Okay, so hello to all of you. So what we're going to be talking about today is protein synthesis. And I'm going to go through this in two different phases. I'm going to go through transcription, which is kind of creating the temporary version of the instructions, and then translation and reading those instructions. All right. Now I want to start with kind of a very important idea. If you remember before, one of the questions I had you on your quiz is, why is protein, or specifically enzyme, under the ultimate control of reactions? The idea is that enzymes control all reactions because you're always going to have the reactants there. You're always going to have, the CHNOPs, all their different elements. And what controls on whether or not we're going to use these elements to create something is the presence or absence of an enzyme. If an enzyme's there, the reaction's going to proceed. If the enzyme's not there, or if it's deactivated, using like allosteric deactivation or inhibition, then it's not going to work. Right? Now, before we said that proteins are under the ultimate control. Well, what's really under the ultimate control is DNA, okay? Because DNA codes for proteins. Now, anytime you want to have a reaction inside your body, and think about this, you're having reactions constantly. If I want to make a cell, I'm going to have to do reactions. So as I grow, or when you were first originally one cell, okay, and then you started becoming 2 cells, then 4 cells, and 8 cells, and so on, you had to perform reactions. And all of those reactions were performed because you had a catalyst there. Well, who made that catalyst? Well, DNA did. Okay? So DNA is really under the ultimate control because they make protein. And protein control, because proteins are enzymes, they control all reactions in the body. So the instructions on how to take carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur, the CHNOPs, on how to take all of those elements and reform them into things like your eyeball or your skin cell or your hair cell is all done under the direction of DNA. Because DNA is going to tell what protein to make, and then protein is going to tell the reaction to take place or facilitate the reaction through an enzyme. Right? So DNA is really the ultimate control of everything. Okay? It's the instruction manual on how to build you, so how to build a cell, okay, which we kind of talked about in our first unit, and then it's also the instructions on how to run a cell, which we kind of talked about in our second unit. Right? So what I'm going to go through today is I'm going to start with creating the transcript. And the idea of creating a transcript, and this is the general gist of things, I'm going to try and use an analogy here, is you look at DNA as kind of the ultimate book. It's our instruction manual. Okay? Well, that book, we can't have a mistake happen in that book. The book's got to be perfect. So we leave that book inside the nucleus. That's our DNA. Okay? But whenever we want to make a protein, we don't need to have the entire book. We only need to have a couple pages out of that book in order to have the instructions on how to, say, make a protein for hair color or eye color or whatever. Okay? So we're not going to take that all the protein synthesis to make a protein is going to happen in the cytoplasm. But the book is in the nucleus. So we're not going to take that book out of the nucleus into the cytoplasm to make a protein. What we do instead is we find a couple pages, make a photocopy of those pages, which we call mRNA, and then have the mRNA leave the nucleus, go in the cytoplasm, and then give instructions. We never have DNA leave the nucleus. Okay? It's too important. And we never actually directly make a protein from DNA. We always indirectly make a protein from DNA by using another molecule called messenger RNA, and that's kind of the link between DNA and the actual protein. Okay? So let me go through my first lecture here, and it's going to be on transcription. Okay, so here's the general idea that we're going to be doing. And today, or our first lecture here, is going to be from DNA to mRNA. Okay, and that's what we call transcription, and that's the title of today's lecture. Later on, we're going to take this and turn it into protein, and that's called translation. But right now, we're making the transcript, which is called transcription. So first thing we want to talk about is how we know that DNA doesn't directly make protein, that there's this in-between molecule. Well, how we know this is there's a scientist, Frederick Sanger, and he showed a couple things. One, he showed that proteins are simply linked of amino acids. So here's a protein, and here's a whole bunch of amino acids all linked together. It's called the polypeptide. We know about this from our first unit. Okay, on what proteins are. They're just a series of amino acids. Now, he also showed, and this was pretty important, that each protein has its unique sequence of amino acids. So if this was insulin, okay, it's not the full insulin molecule, but if this was insulin, the orders of these amino acids has got to be exactly the same for every insulin molecule, whether you're in me, whether you're in a cow, whether you're in a donkey, whatever animal. We all have the exact same sequence of amino acids to make our insulin protein. Okay? If you had a different sequence of amino acids, Okay, and say your glycine was replaced by, say, a valine. Say you replace this glycine with a valine, then this protein would be non-functional, and you would be, say, diabetic because your insulin protein isn't working properly. Okay. So the next step that we need to know was how exactly DNA codes for the production of something like this, and we realized that it doesn't do it directly, it does it indirectly. Okay, and this is how we knew that. Well, we looked at the location of these things. We knew that the cytoplasm is where protein synthesis takes place. But we know that DNA is inside the nucleus. Okay? And we notice that DNA never leaves the nucleus. So how is DNA going to make protein synthesis if it never leaves the nucleus? Therefore, there must be some kind of molecule that transfers the information from the DNA into the cytoplasm in order to make protein synthesis. And that was our discovery of mRNA. Because we noticed that another molecule, similar to DNA, RNA, could be in both the nucleus and the cytoplasm. So we're seeing RNA in both places when we study the cell. Okay? So that's one indication that RNA could be the indirect molecule. The other big thing was we noticed that when RNA levels were high in the cytoplasm, protein productions were high. So when we were making a ton of protein, we had a ton of RNA. When we made no protein, we had no RNA. Okay? So they hypothesized that maybe RNA is the link between DNA and protein, and it is. Okay, it's the messenger between DNA and then the eventual production of protein. So researchers began researching a specific RNA molecule and concluded this type of RNA acts as a chemical messenger for DNA. And we called this RNA messenger RNA, or for short, mRNA. Okay? So further research shows that mRNA attaches to ribosomes to make protein. mRNA is temporary and disposable. After we're done, we just throw it out. Okay, it's not like our DNA, which we never would throw out. We always keep it together. mRNA is disposable, just like a photocopy is disposable. An mRNA sequence is complementary to the DNA sequence. Okay, so we find the DNA from the coding strand. We make an mRNA molecule, which is complementary to it. Okay, so cracking the genetic code. Once mRNA was understood to be the transcript to make protein, researchers began to decipher the code. Okay? And they wanted to figure out, well, out of the mRNA sequence, how does it actually create an amino acid? And they were trying to figure out, well, is it one base for every amino acid? And they said there's no way because we only have 4 bases. And we have 20 amino acids, so 4 bases, if it was a 1-to-1, wouldn't be able to make 20 amino acids. What if we used 2 bases? Okay, well, if we used all the different base combinations, A and T, A and G, G and C, G and T, all of those can only make a maximum of 16 amino acids. Well, we have 20. So maybe it takes 3 bases or a codon to make an amino acid. And they figured out that with 3 bases, you can make 64 different amino acids. We don't need to have 64, we only need to have 20, but that's how we make our amino acids. It requires 3 bases. Right? So this is the information here. We take our DNA, okay, here's our DNA template strain, we're going to make our mRNA, and then every 3 bases is considered a codon, and 3 bases together codes for 1 amino acid. Then the next 3 bases codes for another amino acid. The next 3 bases codes for another amino acid. Okay, and here's our universal code. So you're going to have to print one of these out. Make sure you have one printed for the exam, because I'm going to ask you how to use this during the exam as well. It's probably your next test. And how this works is basically here's your first letter. So if I had UAC, so I'm going to code this UAC. Well, in order to use the table, my first letter is a U, so I go to this box right here. My second letter was A, so I'm going to go over to this box. And then my third letter was C. So I've got UAC, and that's tryptophan. And I go back, and that's TYR. Okay, my next one is going to be GAG. And if I go to my sequence here, my first letter is G, my second letter is A, my third letter is G, GAG, glutamine. Okay, so that's how we use that thing. Now there's a couple things I want you to really pay attention. Okay, if you notice the AUG, methionine, got a special color. All proteins, especially in eukaryotics, start with methionine. It's the first amino acid of all proteins. So you're going to have methionine, and then you can have any other amino acid sequence. But the first amino acid has got to be methionine. Also, there's a couple codes here to stop protein production so that you've completed the protein. And now we have to tell the ribosome that we're done the protein. So we have some stop codons here as well. Okay, so let's make the transcript. So there's a couple videos here. I've already asked you to watch those videos. Make sure you look up some videos on how to make a transcript. So recall, like mentioned before, DNA is incapable of creating protein directly. Okay, so we're going to use mRNA as our messenger between the DNA and the protein. So DNA requires an intermediary to transfer the message from the nucleus to the ribosomes. And this intermediate is a special type of RNA called mRNA. So there's some differences between DNA and RNA. Okay, we've already gone through this when we went through macromolecules, but this is just kind of a reminder. The big things here is knowing that there's different types of sugars, the different bases, U instead of T. RNA is single-stranded. The location, DNA is only going to be in the nucleus. RNA is in both. And DNA is non-disposable. RNA, we make copies, we destroy them, make copies and destroy them. It's disposable. Okay. So just looking at RNA, we're going to talk about a couple different forms of RNA. We're going to have mRNA, tRNA, and rRNA. Okay, we've got a couple different forms. The mRNA is messenger RNA. tRNA we're going to talk about when we do transcription. And then rRNA we'll also talk about when we do transcription. Okay, so overall, transcription, it's the making of an mRNA molecule. It's to create an accurate copy of the small section of DNA. It occurs in 3 phases: initiation, elongation, and termination, just like how replication does. So we'll start with initiation. Recall that only certain segments of DNA are considered coding sections, and we call those exons, and other sections are called introns. Therefore, it's important when making an mRNA to ensure you have the proper code sequence. The following is a list of steps involved for the creation of an mRNA molecule. The first one, DNA contains a double helix with different codes on each side. The first step is to determine which side is going to be the template for RNA. So we've got double-stranded DNA. We've got to decide, well, which side of this is going to be the template. Okay? Now, we call the one that actually is going to create the mRNA a template, or it's also called an antisense. These are the 2 names for it. Okay? The other one that doesn't create the mRNA, okay, has a very poor name. It's called the coding strand, which is very confusing because you're not actually using that as the coding strand, or the sense strand. Okay, so the one that actually makes the mRNA, it's called the template or antisense. The side that doesn't is the coding or the sense strand. Okay, I know that's confusing, but I didn't make up the names for these things. Okay, so we are going to take our template strand, and our template strand or antisense is going to create our mRNA. It's going to be complementary. If you notice, the mRNA is the exact same as the coding, right? Because this mRNA is going to be the complement of the antisense, okay, or the template. The only difference is the T's will now be used because it's RNA. So the first thing we're going to have to do is we're going to figure out exactly which one's going to be. Is it going to be this top one we're going to copy or the bottom one? And say we want to copy the bottom one, so that's going to be our template. Okay. Next thing, we're going to open up this DNA. Okay. Now, in order to open it up, okay, we need to find an area that we need to start in. That's always called the promoter. So the promoter is an area that's going to start DNA creating the transcript. Okay. So DNA is unwound and separated by RNA polymerases. Okay, just call it RNA polymerase. Notice it's not DNA polymerase, it's RNA polymerase. RNA polymerase knows which strand to bind to and where to bind to because of the promoter region. So RNA polymerase is going to come down, it's going to bind to the promoter region. Promoter regions are upstream from the actual gene sequence. So here's the actual gene sequence, the promoter is just before it. And it's going to be a whole bunch of A's and T's. And we should know why there's a whole bunch of A's and T's. Okay, because if we're going to rip open DNA, we want to do it where there's only 2 bonds. And 2 hydrogen bonds are between A and T. There's 3 between G and C. So we want to pick an area that's very easy to open up, and that's where there's a whole series of A's and T's. Okay? And that's what I have down here. We call it a Tata box oftentimes. Okay? And that's going to be the promoter region. So we got a promoter region. It's going to start unwinding the DNA, and then we're going to start making our transcript right in this area right here. Transcription start site. So we're going to start making our transcript here. Okay, so that's the initiation. The next part is going to have to start adding our bases in order to make our mRNA. So here, look, we got our strand opened up, and here's our growing mRNA strand, okay? And we're starting to basically create the mRNA. And you can see the uracils in there and replaced with thymine, and it's complementary to the template strand here, or the antisense. Okay, this is not doing anything here. It's just there for the ride because it's the coding or sense strand. It's not creating anything. Okay. Now, also notice that just like in replication, RNA is made in the 5' to 3' end because RNA polymerase works in the 5' to 3' end. So we're making the new strand in the 5' to 3' direction. Okay, and the template strand or the antisense would be in the 3' to 5' direction. So as soon as RNA polymerase has moved past the promoter and is working downstream, transcribing DNA, another RNA polymerase can bind to the promoter region right behind it, making another mRNA. So we can make the same RNA, more than one mRNA at the exact same time, where I can have a whole bunch of them being made at the exact same time. Now, this combined with the fact that mRNA doesn't get proofread, it allows the body to very quickly make our mRNA. And 2 copies can be made simultaneously very, very quickly. Okay. So once we've created this code, there's going to be a port where we have a stop sequence when it says basically stop making any more protein. Okay? And that's going to be right on our code if we go back to our code here. Okay. You're going to have these 3 letters or these 3 letters or these 3 letters. And as soon as these hit the ribosome, it's going to stop making protein. Okay. And that's our termination. So RNA polymerase continues to add RNA bases until a stop sequence is recognized by the RNA polymerase. Okay, when a stop sequence of nucleotides is read from the DNA, RNA polymerase releases, DNA reforms, mRNA is now complete. Okay. Now, we have to do some modifications to the mRNA. Okay, so I'm not going to go through this part on my lecture because I think the actual content does a better job. So once we have our mRNA, we're ready to basically go into translation, but we have to do a couple things first. Okay? Before we go into translation, we have to take this mRNA that we formed to the transcription and modify it slightly. And the modifications, there's 2 main things that we want to do, and it's right here in your content section. Okay? The modifications of the mRNA. Before mRNA leaves the nucleus, we have to basically add a 5' cap to the end. That's basically a whole bunch of guanines. Okay? And then to the 3' end, we're going to add a whole bunch of A's, which we call a poly A tail. And we do this just to stabilize the RNA molecule. The mRNA will start to degrade inside the nucleus if we don't add this cap and tail. And we want to make sure this mRNA doesn't degrade because it's going to eventually code for the protein. So we stabilize it by adding a cap and then a poly A tail. And you should know that the 5' end is the cap and the 3' end is the tail. And the tail has a bunch of A's and the 5' end has a bunch of G's. Okay, so that's one step we have to do. The only other step we have to do to make sure that we modify our mRNA, and before it's modified, it's called pre-mRNA. And then the last step, so we got to basically add a tail and a cap. Once we add the tail and the cap, the other thing we have to do is cut out areas that don't code for anything. And those are called introns. So here would have part of the code for the protein and the exon. Then we have a break which codes for nothing. Then we have the rest of the protein. And then we have a break in code. And then we have the rest of the protein. We've got to remove all these areas that don't code for anything. And we call those introns. So we use this thing called the spliceosome, which basically removes all of the introns and puts all the exons or the coding regions together. And then we have a true mRNA molecule, and we're ready to go through translation. Okay? So make sure we understand those modifications. So transcription is the creating of the pre-mRNA. Then we're going to modify the pre-mRNA in the nucleus into mRNA. It now leaves the nucleus, and we can go through into translation. Okay, so watch my lecture on translation. I hope this helped, and ask me any questions through email if you have any.